Welcome to This Explainer. Today, we're diving straight into the fascinating microscopic world of exogenous peptides. If you've ever wondered how modern molecular science is actually learning to communicate directly with ourselves, you are absolutely in the right place. We're going to explore exactly what these molecules are, how they work at a mechanical level, and how they seamlessly mimic our native biology to influence literally everything from tissue repair to cellular energy. Here's our roadmap for today. We'll start with the body's native messengers, look at growth hormone secretagogues and tissue repair, jump into mitochondrial energy regulators, explore brain and immune modulation, and finally look at the absolute frontier of peptide science.
Section 1. The body's native messengers. Unlocking cellular communication. Let's establish exactly what we're dealing with at a cellular level.
So here's the crucial thing you've got to understand. When we talk about exogenous peptides, we aren't talking about, foreign synthetic chemicals forcing your body into unnatural states. A peptide is simply a short chain of amino acids. They're basically highly precise biological text messages. And the word exogenous, that just means they're introduced from outside the body. But their entire purpose is to seamlessly mimic the exact signals your cells are already using natively to trigger normal physiological loops.
Think of traditional, blunt pharmacological drugs like a giant master key. You jam it in, it forces open a bunch of doors at once, and boom, you get side effects. Peptides? They're totally different. They are engineered to be highly specific keys that fit perfectly and exclusively into designated cellular locks, or receptors. Because of that crazy specificity, they trigger very precise downstream effects without the unwanted systemic collateral damage.
Section 2. Growth hormone secretagogues, preserving the feedback loop. Let's look at why longevity researchers are so obsessed with this category. Check out this number, 14 to 15%. That is roughly how much our natural growth hormone output drops every single decade after we hit adolescence. It's a biological reality known as somatopause, and as that output falls off a cliff, the downstream signaling that keeps our muscles strong, burns fat, and gives us that deep, restorative sleep, that shifts right along with it. To tackle this, research highlights a really cool tortoise and hare synergy. CJC1295 is our tortoise. It's an analog that provides a nice, sustained, long-lasting release over time. Ipamorelin is the hare, a super selective mimetic that hits the pituitary for an immediate, quick pulse. When you take these two together, you're activating two distinct receptor pathways at the exact same time. It creates this robust, entirely natural pulse of growth hormone that completely outshines what either could do on its own. Now, a massive distinction in the clinical literature between these secretagogues and traditional synthetic HGH is that secretagogues actually preserve your body's natural feedback loop. Basically, if growth hormone gets too high, your body can still step in and say, okay, whoa, that's enough, providing a physiological ceiling. But to be clear, research does note some biological side effects to watch out for, things like transient redness where injected, mild headaches, and maybe some potential shifts in fasting glucose or insulin sensitivity. Interestingly, though, because it's so incredibly selective, ipamorelin manages to dodge the cortisol and prolactin spikes we used to see with older peptides.
Section 3. Tissue repair and regeneration. Extracellular matrix remodeling. Time to look at the physical architecture of the body.
When we dive into tissue repair in the literature, two peptides totally dominate the conversation. BPC-157 is basically your local construction crew. It upregulates growth factors to literally build brand new blood vessels, and it's highly targeted for tendon, ligament, and even gut mucosal repair. TB500, that's the systemic supply chain. It regulates a structural protein called actin, keeping it mobile so that cells and stem cells can migrate exactly to where they are needed most in the body.
When you combine these two, you get what researchers famously observe as the Wolverine stack. It's wild because they perfectly cover complementary stages of the biological wound healing timeline. During the first few days, the initial inflammation phase, TB500 calms systemic cytokines, while BPC-157 improves local blood flow. Then, in the proliferation phase over the next few weeks, BPC-157 drives the specific cells that synthesize collagen, while TB500 builds out the vascular network. Finally, in remodeling, they team up to organize the tissue and minimize scarring. It is a beautifully synchronized biological dance.
Now, while they're deeply effective at accelerating recovery in preclinical models, researchers neutrally track very specific safety signals. For BPC-157, observations include mild gastrointestinal discomfort and some occasional injection site bruising. For TB500, preclinical data notes transient headaches in roughly 3 to 8% of cases, plus some mild redness. So overall, the biological safety profile is generally well tolerated, but these signals are strictly monitored in the literature.
Section 4. Mitochondrial energy regulators. Intracellular signaling. Let's zoom even deeper, straight into the actual powerhouses of our cells.
We're looking at a peptide called MOTSC here, and it does something truly incredible. Unlike almost every other peptide which comes from our nuclear DNA, MOTSC actually originates from mitochondrial DNA. And when you're under metabolic stress, like during a really intense workout, it performs what's called retrograde signaling. It literally travels backward from the mitochondria straight into the cell's nucleus, binds directly to your DNA, and upregulates protective genes. It's basically an internal distress signal that optimizes cellular metabolism on the fly.
Because this fundamentally shifts your cellular energy metabolism from the inside out, clinical observations show MOSC requires an insanely precise low-frequency protocol. We are not talking about a daily shot here. Research protocols use 5 milligrams exactly once every 5 days for just 4 total doses. Why? Well, because that cellular signaling cascade needs time to actually propagate. Plus, it requires a strict 4-month minimum rest period to let those profound mitochondrial adaptations consolidate. It's fascinating.
So, having explored muscles, tendons, and the intricate world of mitochondria, it naturally raises a really fascinating question. What if we could take this exact same lock-and-key precision, bypass the blood-brain barrier entirely, and speak directly to our neural networks?
Section 5. Brain and Immune Modulation, the Neuropeptide Network. This brings us to a highly specialized class of peptides.
A prime example of this in the scientific literature is Cmax. It brilliantly shows how slightly tweaking a peptide structure, in this case just adding a specific amino acid sequence so it doesn't get broken down by enzymes, allows it to slip right past the blood-brain barrier. Delivered intranasally, it literally signals your brain to ramp up the expression of BDNF, or brain-derived neurotrophic factor. This is huge because it supports neuronal survival, boosts dopamine synthesis, and dramatically enhances synaptic plasticity and learning.
But this neuropeptide network actually extends way beyond cognition, coordinating with systemic immune and barrier defenses too. Take melatonin too. It interacts with receptors that influence both skin pigmentation and your appetite. Then you have thymosin alpha-1, which acts as this incredible immune balancer, promoting T-cell maturation. LL37 is especially wild to me. It's an antimicrobial peptide that literally tears apart the lipid outer layer of bacterial cell membranes. And then there's KPV, a powerful anti-inflammatory that stabilizes the mucosal barriers right in our gut. Section 6. The Frontier of Peptide Science, Mastering Endogenous Feedback. Let's take a step back and look at the big picture.
The overarching takeaway from all the clinical data we've looked at today is that exogenous peptides represent a massive, fundamental paradigm shift. We are officially moving away from the era of blunt, small molecule interventions that carry all those heavy systemic side effects. Instead, we're entering the age of targeted biomimetic therapeutics. We are literally using the body's exact native language, the precise amino acid sequences it already knows and recognizes, to prompt the body to repair, optimize, and protect itself.
Which leaves us with a pretty profound realization. As we continue to map out and decode these native messengers, we really have to ask ourselves, are we simply finding new ways to treat injuries, or are we finally learning to speak the very language of cellular time, energy, and repair? The science strongly suggests it's the latter, and honestly, it fundamentally changes how we view the physiological destiny of the human body. Thanks so much for joining me on this explainer, and keep your curiosity locked on to the science.